Happy Bio students. Today we are going to continue 7.2 and we are going to look at how nucleosomes can help regulate transcription in eukaryotes. So let's remember that eukaryotic DNA is wrapped around histone proteins to form a compact nucleosome. So here you see a picture of your nucleosome. In your PowerPoint notes, the nucleosome looks like this. It's pretty much the same thing, right? The octamer made up of 8 histone proteins, or 2 copies of 4 different types of proteins, and then your histone H1 protein that sticks out here at the end, and this is what allows the DNA to kind of link here with other DNA, and it allows for further packing. So if we look at the nucleosome here, you see the same thing. You see the octamer here in the middle in pink, and then you see the histone protein here. But something very important about nucleosome structure is that they also, the histone proteins, the pink ones, they also have these protruding tails. And these protruding tails are going to determine how tightly the DNA is packaged. Now these tails are positively charged, so since DNA is negatively charged, the tails kind of associate tightly with the negatively charged DNA, and it keeps the DNA tight. So here you see tightly packed DNA. Okay. Now if these tails are acetylated, that means you add an acetyl group to these tails, then that means that these tails are not going to be attracted to the DNA as much. Okay, so that's going to interfere with the attraction between the tails and the DNA. So then that means that DNA becomes loosely packed. So notice here that the DNA is not as tight. Now, what does that mean? If DNA is not tightly packed like so, if DNA is loose, we call that type of DNA euchromatin. That means that since it is loose DNA, those regions that are loose within DNA, they have high transcriptional activity. So that means that more transcription is going to happen. So here you see the tails are acetylated. So adding an acetyl group to the histone tails is going to loosen up the DNA and form this euchromatin. Okay. When the DNA is supercoiled and is not accessible for transcription, then we call it heterochromatin. So that means that the DNA is tightly wrapped around. So how does DNA get to be this way? Well, notice here that on the tails you can also methylate the tails or add methyl group. So whenever tails are methylated, then that means that the DNA becomes very tight, and then that means that there's regions with low or no transcriptional activity. Okay. So you can have different regions of DNA within a chromosome, and this is going to determine which regions of the chromosome are actually can actually get transcribed, or where there's more transcriptional activity. Okay, so notice here that the tails are methylated, right? But you can also methylate the DNA itself. So let's look at one of the skills, which is analysis of changes in the DNA methylation pattern. So it says here that direct methylation of DNA as opposed to the histone tails can also affect gene expression patterns. So here in your notes, you looked at both acetylation and methylation. And here, let's look at methylation. Here you see how the actual DNA structure is methylated, right? So you're adding this methyl group to the actual DNA and not the histone proteins. So what does that mean? Well, we said that the DNA becomes tightly packed, right? The DNA forms this heterochromatin. But also increased methylation of DNA decreases gene expression, right? Because if you can't undergo transcription, then that means that that gene is not going to be expressed into a RNA strand and then into a protein. So what actually happens when DNA is directly methylated is that it prevents the binding of transcription factors. So remember from before, we said that RNA polymerase needs to bind to transcription factors to properly synthesize an mRNA strand. So if the DNA is methylated, then that process is going to be inhibited. So genes that are not transcribed tend to exhibit more DNA methylation than genes that are actively transcribed. So what are some of the factors that contribute to DNA methylation pattern? Well, sometimes it's just fertilization, right? Parent of origin and imprinted in genes. That it's just some type of genetic control. It's studied that maternal diet in pregnancy also leads to some form of methylation. Infancy, early life, exposure to microbes, your life as a young adult, so your environment, your diet, your lifestyle also affects methylation patterns. And as a senior, also age-related changes. So these methylation patterns aren't always genetic, right? It could be changes in your life or what you're exposed to in your life that will actually methylate your DNA. So here we're talking about things that will affect your protein synthesis, right? So this is kind of strange to think that your environment is affecting your DNA and it's affecting what proteins actually get made. But it's actually a very interesting field of study, and we call this field of study epigenetics. So it's a study of changes in phenotype as a result of variations in gene expression levels. And these gene expression levels are a direct result of DNA methylation patterns over time. Like I said before, it is influenced by heritability, but it's not genetically predetermined, right? Not all the time, and we can see it because identical twins may have different DNA methylation patterns as they grow older. So here we see a comparative DNA methylation pattern in twins of different ages, and it's actually really interesting to see what happens here. So here you see the methylation pattern in twins when the twins are 3 years old. And if you notice here, they have similar levels of methylation. So they're about the same. So in this graph, it shows you the number of different bands, and notice that there's not that many different bands. There might be some changes here and there, right? But it's mostly the same. And that's because if you're a 3-year-old, you probably have not been exposed to any of this stuff, right? Yes, you were inside your mother's belly, and you've been through infancy, but you have not been through these environmental life events and factors and diet and lifestyle, right? But notice that the study done in 50-year-old twins, notice here, here, and here, and here, how the methylation patterns are different, right? So there's a lot of methylation, and here in this bar graph, it shows you a high level of differential bands. So they have different methylation patterns, and that's because even though they're twins, right, and they do have the same DNA, the DNA has been methylated within itself in different patterns. So you end up with different differential bands. So the findings is that DNA methylation patterns differ between clones or twins and change over time. And methylation patterns have a genetic basis but are influenced by the environment. Here we see another study that was done, a comparative methylation pattern in twins of differing health status, healthy versus diseased. So here you see an affected twin on the y-axis and a healthy twin on the x-axis. And here you see differentially methylated probes. So this is showing you that DNA methylation patterns differ between healthy and unhealthy clones. notice that the affected twin has a greater range of methylated probes in comparison to the healthy twin. So then that means that methylation controls transcription and hence phenotypic expression of a disease. So in some way, this disease has been affected by the rate of methylation of this twin, and it has controlled transcription in some way, and it has controlled the phenotypic expression of the disease in some way. So methylation has phenotypic effects at some point. Okay. So in your activity, you are going to look at another example of changes in DNA methylation pattern of some twins, and you're going to try to determine what are some of the changes that you see, and then we will discuss what your findings were. All right, I hope this video helped. Have a good day.